Hello, good afternoon. We are going to basically do a little bit of review today for mostly lecture 7. I mean, if you have questions for earlier lecture, for lecture 6, you can also ask that today. But we will spend about an hour doing that. Afterwards, I will be going through the lab with you, so you know what to do for the microscopy lab. So, let's get started. Here we have, a strand of DNA, and we can represent it using just like a rectangular box. But know that in the rectangle box is just continuing having the DNA, right? And then it's gonna go on like this. Okay. So this is DNA. And of course, the DNA, there are many genes in it. Right? So this would be gene number 1, gene number 2, gene number 3, which is just a section, right, of the DNA that contains the instruction to make proteins. So each gene has a beginning sequence. Does anybody remember? What do we call the beginning of a gene? It starts with a P. No? That's all right. That is the promoter.
Water.
Yeah, thank you. Promoter, which is
Region.
the beginning of a gene. And as you may recall, it is AT rich. Okay? Beginning of a gene. We also have a sequence at the end. Right? And that is called the terminator sequence. Terminator is the end of a gene. All right. Now, I mean, maybe I should just do this in a different way. The gene doesn't have to be like, right next to each other. Right? It's possible that, you have some filler DNA in between before you get to the next gene, right? So it's just a segment of DNA. Now let's say we want to copy gene number 1. And we'll copy gene number 1. So we're gonna do that in the process of transcription, right? Transcription. And so we would open up the DNA. Right? This would be like the beginning, right? The promoter. And this would wind up into the DNA, but I'm just gonna keep it simple like that. So we're not gonna get too confused. It's gonna be 5' to 3' on the top, and then 3' to 5' at the bottom. And then you will have the terminator here. So we're gonna have an enzyme that's gonna help us with transcription. What is the name of this enzyme? Does anybody remember? To build our messenger RNA. What enzyme do we need for that?
The RNA primer?
I think you're thinking about the DNA replication, in which case, yes, you would need the RNA primer, but this is, we're not trying to copy the DNA. We're not trying to copy both strands of DNA. We're trying to do transcription, right? We're trying to make RNA. Anyone else? Trying to make RNA. That's fine. That's why we do the review. So you remember, this is RNA polymerase. Okay. So RNA polymerase is going to move along the template DNA from 3' to 5' and make a copy of RNA. So let's put some letters here, nucleotides. Right, so on and so forth. Right? So on the other side, it's going to be U, A, G, C, G, U, A, C. Oops, I'm sorry. There should not be any U there. We're talking about DNA here. I am very sorry. Got confused. There we go. Okay. So up here, this is the coding strand, the coding DNA, which contains the instruction to make the protein. I keep on telling you that. And the bottom strand here, that is the template strand, template DNA. All right, so basically, our let me see if I can do this. Our RNA polymerase is going to move forward. Actually, it won't let me do that. See, it's going to move forward, and it's basically going to, without the TNA, I cannot not drag it, but just ignore the TNA. But this thing is going to move along the bottom, and then it's basically going to build the RNA strand, right? So let me try that one more time. All right. It's going to move along. And as it moves along, it's going to build the complementary strand. 5' to 3'. It's going to keep on going. And at the end, you are going to get your premature mRNA, right? This is not quite ready to go out to the cytoplasm yet. We still have to modify it a little bit. But that's basically the process of transcription. Okay. So we're going to take our transcript and take a look at it. Right? We're going to have the pre-mRNA like this. And it turns out the pre-mRNA has some sequence in it that are non-coding, right? You might remember that. What do we call the non-coding section in a gene? Does anybody remember the non-coding section of the gene? Is it exon or is it intron?
The non-coding would be intron.
That is correct. Thank you.
Intron.
So this is the intron. These are introns, which are non-coding. Non-coding section. Segment, you can say. So we have to remove the intron and join together the exons, right? Exons are coding segment. All right. So a couple things happen here. We have to add a cap on the 5' end. And this was the 5' end. This is 3' end. This is a 5' guanine cap. And what do we add on the other side? Which letter do we add?
The A and T, on the end.
Yep, just the A's though. Okay, no T. Just a bunch of A's. Okay, so this is called the poly A tail. So the cap and the tail together will increase the stability of the RNA in the cytoplasm. So after you remove all the exons, you would, after you remove all the introns rather, you can join the exons, and then you will have the 5' cap, and then you will have the poly A tail. Okay. So all these things that you do, these 3 steps, the adding the cap and then the tail, as well as removing the introns, this is called post-transcriptional. Modifications. All right. All this happens in the nucleus. And out here is, of course, the cytoplasm. Once you finish the post-transcriptional modification, this is going to leave the nucleus through the nuclear pore. Okay, we can put a few more pores there if you want. Nuclear pore, nuclear pore, nuclear pores. Okay. So they're going to come out. And we're ready for translation. So there's my sequence again. Gonna have the G cap and the tail. The longer the tail, the longer it will last in the cytoplasm. So let's put some codes on here. I guess I should use the, never mind. I'm just gonna make some new codes. It doesn't matter. It's AUG, UUU, CCU, ACG, UGA, something like that. Okay. I mean, it's supposed to be the same as the strand here, right? But then it's just easier for me to make a new stuff, new strand here. Okay, so this is the messenger RNA, right? Messenger RNA. It contains the code to make the protein. It's the recipe I told you. And then you're gonna have your ribosome here, right? The ribosome, which contains the rRNA as well as the protein. And that's going to be creating the protein, joining the amino acids together. And we also have these things that are flying around. Okay. These are the transfer RNA. Right? Transfer RNA. They look like clover leaves. And each one of them is going to be carrying amino acids. So at the bottom here, we're gonna have some anticodon. Something like that. So the anticodon has to be complementary to the codon. Okay, it has to be complementary to the codon. And so when I give you a tRNA like this, and I ask you, what is the amino acid? What is it carrying? You will need to look up the codon, right? So you have to take out your codon chart. And again, I keep on telling you, you got to print this. You have to print the codon chart and bring it to the quiz, right? On Thursday to do it. So can someone tell me what is the amino acid here? If the anticodon is UAC, what is the codon? Here, maybe I'll bring the chart over. Here's the chart. Okay. What amino acid will this thing be carrying? Look it up and tell me, please.
MET?
MET is correct, right? Remember, we have to look up the complementary one, right? So this is complementary to AUG. Right? So that's MET. That's what this is. MET. Right? So when this one comes over here, oops, I think I didn't pick up the U. Let me try that one more time. If I bring it over here, it's gonna be matched, right? U and A pair up, A and U pair up, C and G pairs up. That's how we know is the correct amino acid, right? So that's gonna deliver it, and it's gonna put the MET right here. Okay. So next one is UUU. If we look up UUU, we're gonna find out it's PHE, and the anticodon for the UUU is AAA. So that's gonna be that one. And so this will come, and then you will have the PHE. And then the ribosome is gonna create the bond that links them together. And so on and so forth, right? So CCU is pro, ACG, it's THR, and then that's it. This is a stop codon. Right? So we did this last time. That's the process of translation. Does anybody have any question? All right. So let's take a look at the study guide here. We should try these tables out. This is question number 6 in the study guide. I'll give you a moment to grab that. So we have to complete the table, and we are given the coding strand. What's the relationship between the coding strand and the mRNA? Well, you should remember that by now. Coding strand and mRNA are actually identical. Identical. But T becomes U, right? T becomes U. So all we have to do is copy it. ATG, I'm gonna copy it. AUG. Change the T's to the U, and that is it. It's pretty straightforward. You just copy everything. Done. What's the relationship between mRNA and anticodon? We just draw it out, right? The mRNA is the strand with the co here, and then the anticodon is the one that is at the bottom of our tRNA, right? So between mRNA and anticodon, they must be complementary. Okay, these were complementary. Complementary means follow the base pairing rule. So A pairs with U pairs with A, G pairs with C. AUA, the anticodon will be UAU. C pairs with G pairs with C, A pairs with U. And so on and so forth. Okay, we don't need this. And this is the subcodon. We can put AUU, but know that no anticodon for stop codon. Right. We'll say technically. Right, but that's okay. We'll put it there. That's the reason why the translation stops, right? When you get to the end, there's no anticodon, and we're not gonna make the protein. We're gonna stop making the protein, and then we're gonna look up the amino acid. Please be careful when you look up the amino acid. We are using the mRNA to look up the amino acid. Okay. Here, use the mRNA to find the amino acid, not the anticodon, right? And you're just gonna look it up. I have my codon chart here. Hopefully you have your own as well. And this is gonna be MET, ILE, and then stop. Do you understand this table?
I have a question.
Mm-hmm.
The mRNA, that's the codon?
Yeah, the nucleotides on the mRNA is the codon. That's correct.
Okay, so
That's the
that
one you look up. Yeah.
okay, so that's used that term is used like interchangeably, mRNA and codon. It means the same thing. It's the same thing.
Well, mRNA is the whole strand, right? So like each of these triplets on the mRNA, we call those the codon. So the proper way of saying it is, I guess, like the codons are found on the mRNA, right?
Okay.
So like let's go back to here. Like this whole thing, the red thing along with the green stuff, that whole thing is a messenger RNA, right? The codons are the other parts here. Like you might have some other letters here, like CCA, TUG, whatever, right? Those are part of the mRNA, but they are not really the codons, right? Does that make sense?
Yes, because I was just thinking back because you said we never look up the anticodon.
That's right.
So then I was like, well, where's the codon? So okay, I get
Yeah,
it. mRNA. Okay.
that's right.
Okay. Yeah.
Good question. Anyone else? Are we together? You gotta know how to do this on the quiz. Let's do one more together then. Over here. We are given the template strand this time. What is the relationship between template and mRNA? Template and mRNA. What's the relationship?
Complementary.
They are complementary. The rest of you know this too, right? Please tell me you do. And if you don't, ask. Ask a question, please. I don't know what you don't know if you don't tell me.
Sorry, sir.
Hi. Go ahead.
One slide back. Can you go back one slide?
Oh, can I go back one slide? to where? Sorry,
So
the drawing here.
yeah, this one, yeah.
Mm-hmm.
This U,
Uh-huh.
are we going to put direct UUU or uh, AAA?
Are you gonna put UUU or are you gonna put AAA on what?
I mean, if it is AAA, it's
Mm-hmm.
not PHE.
That's right. So we're gonna look up UUU because the UUU is the codon, right? How
Yeah.
do I know it is the codon? Because it is on the messenger RNA. Okay. So when do you look up AAA? You don't look up AAA. AAA is the anticodon. That's the anticodon. That's for when the tRNA brings the amino acid over, the anticodon has to be complementary to the codon for it to click.
Uh-huh.
Let's say this one over here, right? This one here has a UUC. Let's say it comes over here. Like UUC is not gonna match with the UUU, right? You understand what I mean? How do I do this? Hold on. Let me let's just erase this one. Okay. I'm gonna bring this amino acid over here. Okay, and then I'm gonna put there. That's not gonna click. It's not gonna fit because the UUC is not complementary to the ACG. You understand that? That's
Yes.
that's the purpose of the anticodon. That's the purpose of the anticodon. Right. So it's just for the cell to know which tRNA is the right one. That's why when we look up the codon chart, we don't look up the anticodon. We look up the codon.
Okay, thank you.
Yeah. ACG was THR. Any other question from anyone else? If you have a question, someone else probably has the same question. Okay. So over here is complementary, which means I have to use the base pairing rule. So TAC is going to become AUG. GGG is going to be CCC. GGA. UAU. GCG. And UAA. What's the relationship between mRNA and anticodon? mRNA and anticodon. Are they the same or are they complementary?
Complementary.
Complementary again, that's right. So we got to flip them again. We got to use base pairing rule again. So UAC, GGG, so on and so forth. That's it. And then we look up the amino acid. MET. You can do this part, right?
Sir, just a question. Can we do like a table for this?
Yeah.
Like the
cheat sheet,
template
right?
coding?
Yeah.
Yeah.
So I wrote this down for you guys last time. Let's do it again. Okay, stick it on the wall when you do the test. So let's start with the, doesn't matter, template, coding. So there are only 4 choices, right? A, C, G, T. Help me out, guys. What is the coding strand? If the template is ACGT, what is the coding? Type it up in the chat box. If the template is ACGT, what is the coding? Yep, I have two answers, and they are both correct. TGCA. Complementary, right? Okay. What about the messenger RNA? Well, messenger RNA is a copy of the coding strand. So it's the same. I just copy the coding. But I have to change the T to U. And then finally, we have to have the anticodon. And anticodon is complementary to mRNA. So it's going to be ACGU again. Okay. So basically, these are the same. And then these are the same. Right? And then the different color ones are complementary to each other. The yellows are complementary to the blue. Right? Mind you, these are DNA. And these are RNA, right? The codon is on the mRNA and the anticodon is on the tRNA. Is everybody okay? You guys are so quiet. I can't tell. I can't tell if you get it or not.
No, sir.
Okay, which part do you not get? Ask me a specific question.
Um, I think I'm a little bit confused between that question 6A and B, because the first one you started with the coding strand, and then after with B you started with the template strand.
Do you know the difference between the coding strand and the template strand?
No.
Oh, no? Okay.
I-
That's fine. Okay. So like the coding strand is the strand that has, when I say the code, right, that's the instruction to make the protein. When I say instruction to make the protein, do you know what I mean by that?
Yes.
Yeah, like it tells you which amino acid to put together, right?
Yeah.
So it makes sense for the messenger RNA, which is the recipe, to look exactly the same as the coding, don't you think?
Yes, but
Because yeah.
should we have started first with the template strand? What
Sorry, what's the question? I missed
can we
the.
have started like with first template, then coding, then
Yeah, you could do that.
mRNA,
Yeah,
then anti, then we get the amino
sure. Yeah,
acid.
that
Yeah.
works too. You know, if you guys are having trouble going from the template to messenger RNA, that seems to be what I'm hearing. You can change it to coding first. Here, I can add a layer. I can add an extra thing here, right? So like, the relationship between coding and template is complementary.
Complementary. Yeah,
That's right.
change
So
it.
that's right. So you can do this if you want.
Okay.
Is that what you're asking me?
Yes.
Yeah, so you can write it out first, no problem. And then you would just copy it to make the mRNA, right? Is that what you're saying?
Yes.
Yeah, that
Just
works.
change the T to the U, right?
Yeah. Change the
Okay.
T to U, exactly. Yeah.
Okay.
So like if you get lost on the test, right, look at this, cheat sheet. Template, right? T becomes A, right? A becomes U. You just, use that as a reference, right? Does that help a little bit? Anyone else? Okay. Now you're not gonna have to type in these things on the test, because it's challenging to type them in, I suppose. But let me give you a question. Like over here. Okay, what do you think? Right, tell me the answer. Yeah, bring the cheat sheet down as well, in case you need it. You know what? Never mind. It's on the other file. Okay, just try your best here and tell me what you think the answers are. Okay. Anyone else? So, sorry, I keep on talking and interrupting you guys. When you do something like this on the test, you shouldn't be looking at the answers. You shouldn't be like trying to match it up and try to figure out which one is correct. That's gonna confuse you and you're likely to make a mistake. You should just like totally ignore the answers. Just completely ignore the answers and do the question on a piece of paper. All right? It says this is a template strand, okay, and you want to make RNA. So what's the relationship between template and RNA? If you're not sure, you can convert it to coding first, right? Or if you remember, they are supposed to be complementary, right? Complementary. So if I see TTA, I'm gonna have you change it to AAU, right? And you don't even have to do the whole thing. You can just do a few letters here. And then you go find AAU. Is that AAU? No, that's not AAU. Let's not choose that. There is an AAU, okay? Let's just make sure there are no other AAU. Not AAU, not AAU, not this. The answer is B. Do you understand? That's how you do it. Okay? Don't look at the answers and then try to figure out which one it is. It's just gonna be like super confusing. All right, one more for you. There we go. Okay, that's good. That's good. Yeah. This is mRNA sequence. I want a tRNA, the anticodon. And what's the relationship? You must be sick of hearing me say this word. They are complementary. So if it's AUG, we're gonna have UAC, right? And some stuff. I mean, you can do the whole thing, but just see, just do a few, and then you should be okay. AUC, nope. AUC, nope. Nope. Okay, that's probably it, but let's just make sure. Okay, that's not it, right?
Sir,
I mean,
on the mRNA strand, there's no T.
that's right, because RNA, right? There's never T on any kind of RNA, be it mRNA or tRNA, right?
Okay, that's
Yeah.
good.
So like I mean, thinking along that line automatically without doing it, you can reject this one, and reject that one, right? Do you agree?
Yes.
Because then we're asking about RNA. Anytime you see a T, it's like that's not the answer. right? So you automatically take out 3. Now you're down to 50-50 as opposed to like, 20% chance. Increase your odds.
Okay.
Okay. It's not too bad. This test, there's no, I don't, there isn't any like multi-select, right? It's just all regular multiple choice. Anyway, I hope this brings you some comfort. Anything else before we review mutation?
Yes, can I just can I restate this and tell me if I'm
Sure,
getting it
sure. Yeah,
correctly?
yeah.
Please, just I keep going back to it because I feel so lost
Yeah, that's fine.
sometimes.
Yeah, go for it.
Okay. Okay, so we have the template and the coding strand, and then that is complementary to each other.
And
The
they're on the DNA. Yeah.
Yes,
Mhm,
so that's complementary. And then the template and the mRNA,
mhm.
that is also complementary,
That's right. Yeah.
right?
Yeah.
Okay.
That's
Okay,
all.
and
Yeah.
then okay, sorry. And then the mRNA
Mhm.
and the anticodon is complementary.
That's right. That's right. So
Okay.
it's if you go back to this thing here, uh like the
And then the mRNA and the template is the same.
uh the mRNA and the coding are the same, right?
Okay, sorry, mRNA and coding is the same.
Hmm.
And what about the mRNA and the template? That's
Complementary. You said it yourself, right?
oh
Complementary.
yes.
Yeah. So
Yes.
look at this one here. Same color means the same. Different color means complementary. Okay. I am not kidding, guys. I'm not trying to be like condescending or anything. Write this down, because it's easy to get confused on the test. Write this down, highlight it exactly the way I have it, and then use it on the quiz. It's fine, right? Like you sometimes just need to jog your memory a little bit, right? Okay. So template and coding complementary. That's why they're different colors. Okay. mRNA anticodon complementary. Coding strand and mRNA, they are the same. And when I say the same, you got to be careful, right? Like obviously the T are replaced by U, right? You should know that by now. And then template and anticodon are the same. Are we okay? All right. We are almost there, guys. 3 more lectures to go, and then that's it. Okay? That is it. You are almost done the course. And unless you end up in my class again next semester, otherwise you won't hear from me again ever. So just hang in there. Okay, let's take a look at number 8. If a DNA sequence contains 360 nucleotides, how many amino acids do you expect to be in the protein sequence? What do you think? Give me some numbers. Is it 360? Is it 1080? Is it 120? Is it 36? Or is it none of the above? Okay. Anyone else? All right. Can you guys explain to me why it is at 120? Just so I know you know why, and it was not a guess. Why 120? Yeah.
I
Go on.
can
Hmm.
Each amino acid has 3 letters. So then
Nucleotides, yeah.
yes, nucleotides. So then it's 360 divided by 3.
That's it, right?
It's
You need
grouped in 3.
exactly. It's grouped in triplets, right? It's grouped and codons, right? So if you take divided is 120, right? Assuming no introns. Okay, because this is in DNA, right? So assuming we're not cutting anything out, the maximum number of amino acids you can encode for is 120, okay? Excellent. All right, let's take a look at this thing here. I know it's hard to tell when it's black and white, but you know, let's make do with what we have. Over here, we have a base pair substitution, and the U is replacing the A here. But that's okay, because there are no change in the amino acid that it's coding for. So what do we call this? Silent or neutral, right? Mutation. No change, no effect. Best kind of mutation to have. Actually, you know what? Let me just circle it so you know where the changes happen. Over here, the C is replacing the A, and as a result, the threonine becomes the proline. So we have a different amino acid. What do we call this? When the mutation gives you a different amino acid in the end, what is the name of the mutation?
Missense or
That's
mistake?
right. Yes, it's a mistake. That's why it's called missense. Very good. Okay, so you would get this kind of question on the quiz. I will give you a sequence, and then I'll give you a mutated sequence, and then I'll ask you like, what's wrong with it? Right? What's the mutation? Maybe it's silent, maybe it's missense, maybe it's other stuff. Okay, so over here we have a double base substitution, changing the A and the C here to U and A. And of course, UAA is a stop codon, and that gives you a nonsense mutation. Nonsense mutation. And then the whole thing is missing, right? You have a truncated protein. Not a good thing. Over here, we have a deletion. Okay. And then the deletion leads to a frame shift. You shift the reading frame, and as a result, everything is going to be wrong after that point. Right? We're going to have extensive missense. Okay, everything's going to be wrong after that point. Next one here, we have an insertion. You're putting in extra things. And in this case, the insertion created a stop codon that wasn't there before, and therefore is nonsense. Right? So you can create nonsense mutation by insertion, by deletion, as well as base substitution. Over here, we lose a codon because we are deleting in multiples of 3, right? Deleting multiple of 3, no frame shift this time. And then you are just missing one amino acid. Okay, it might be okay. It might be fine because you're just missing one amino acid, right? Might not be a big deal. All right.
Sure, just a question.
Mm-hmm.
For the last one that you did, the deletion, the no frame shift, because nothing is really happening except that it's missing, can we also say that's like silent or neutral?
It's not silent. It's not silent because you're missing an amino acid, right? So it's still gonna possibly have an effect, but the effect is not as great. right? So like depends where the amino acid is missing. If it's missing in a key part of the protein, then you could cause the protein to not function, right? Like we talked about the enzyme last time. If you're missing an amino acid in an active site, sometimes that's enough to cause the protein to malfunction. But if you're missing it in, some non-essential part, um, then then maybe it's gonna be okay. Does that make sense?
Yes.
All right. So there's this question that we didn't do last time. Let's take a closer look at it. Which of the following mutation is likely to be the most deleterious? Deleterious means harmful. Okay, so which is the worst? All right, so let's see. Deletion of AAA at the beginning of a gene. Well, if you delete AAA, you're gonna be missing an amino acid. Okay, like I said, it depends on where that amino acid is. It might be problematic. But we are looking for the worst outcome here in this question. So, let's take a look at the other ones and see, if there's anything that's worse. Okay, UAC is mutated into UAU in the middle of a gene. Can we look up UAC, please? What is UAC? Where is the codon? Okay, tell me when you have UAC. Take a look at your codon table. Okay, thank you. UAC is TYR. What about UAU? What is the new codon? What does it represent? Yeah, also TYR, right? So do you think this is deleterious? Do you think this is bad? Hopefully
No.
no. Yeah, no, right? It's the same thing. It's silent. This is neutral. No problem. Okay, not harmful. Next one. UAC is mutated into UAG. Well, UAC is this TYR thing. We looked it up in the previous choice. And then it mutates into UAG, and hopefully you remember UAG is stop. Okay. Do you think this is bad? Near the end of a gene?
No,
Why not?
because it's just near the end of the gene and it's gonna stop anyway. So
Exactly, right? It's gonna stop anyway. So we will lose a little bit. Maybe we will be okay. Next one. Deletion of an A nucleotide, A as in like adenine, right? At the beginning of gene. Right? So this could be bad because you will have frame shift, and as we have seen, when you have a frame shift, everything is wrong after that point, right? You're gonna get a completely different set of codons. Your amino acid is gonna be different. You're gonna be making something totally different. That's pretty bad. I'm gonna go with that one. But let's just check the last one. Insertion of UAA, which is a stop codon, at the end of the gene. This is basically same as C, right? You're stopping, but near the end, you're gonna stop anyways. So maybe it's okay. That's why the worst is gonna be D. Is this okay? All right. So that is it for the review for lecture 7. Don't forget you still have to study lecture 6, but I think lecture 6 is much more straightforward compared to 7. You gotta know your mitosis, you gotta be able to identify them from diagram, and then there's the DNA replication, right? The enzymes, the helicase, primase, all that stuff, as well as the experiments, right? Okay, you still have time to study for them. Anything else you want to ask before I talk about the lab?
yes.
Yes, go ahead.
I just wanted, because at the beginning of this lecture, we were talking about the RNA polymerase,
Mm-hmm.
right?
Yeah.
Um, but in the first, I think like maybe lecture 6 when
We're
we
talking
did
about
the,
DNA polymerase.
yes.
Are you confused between the two?
Just a little bit. Like I know like the RNA will be different because like that's different from DNA. So that's why there's DNA and RNA polymerase. But I just wanted to know more of
Yeah.
like a comparison.
So the DNA polymerase 3 and 1 are used for DNA replication. Okay. When do you do DNA replication? DNA replication happens during interphase, right? And it is necessary for mitosis. Mitosis is the splitting of the cell, right? Before they split, you have to copy all the genetic material. Copying all the genetic material, that's called DNA replication. Since you're building DNA, you need DNA polymerase. Okay, in case you missed it, polymerase it spells polymer and then ASE. ASE means it's an enzyme. This is an enzyme that builds DNA polymer. Right? DNA is a polymer. You know that from lecture 2, right? It's polymer of nucleotides. So that's DNA polymerase 3 and 1. On the other hand, we have RNA polymerase. This is, again, RNA polymer and then ASE. It's an enzyme that builds RNA polymer, builds RNA basically. When do we build RNA? Well, we need this. We need this for transcription, right? For transcription. For
So that's in that elongation period.
yeah,
Correct.
initiation, elongation, and termination. That's right. Right. Needed for making messenger RNA. Okay, so the purpose of RNA polymerase is to make a copy of DNA so that you can use it to make protein. You're not copying all the DNA. DNA polymerase, on the other hand, you are copying all the DNA because you need all of them to be passed on to the next cell, right? When the cells split, you need each half to have the exact same genetic material, which is why you're copying everything for DNA replication. Okay, so here, only copy the section of DNA that is needed. Okay, does that clear it up a little bit for you, or I made it worse? It's always
No, you're okay. Thank you.
all right. Anyone else? Ask away. If there's anything else that's unclear. Fantastic. Let's do the lab, okay? I want you to do very well on the lab this time. Last time, I mean, it was okay. Most of you did all right, but then, it wasn't the greatest. So let me try to help you even more for this lab. So to get the lab, you're gonna go to your section. Okay, this is 201. That's okay. You can go to 202. Go to content. Okay, content. Virtual labs, and then you already did the beetroot. So now we're doing the microscopy. Okay, and then you want to download this file. Okay? These two links, it's in the document anyway. So just download the document, okay? And it's gonna look like this. Lab 2 microscopy, 10% of your final grade. Okay. Watch how to use and care for a microscope. So you click on that, you watch that. And it's really short video. Visit this link. Right? This is important. Right? You're gonna click on it, and it's gonna show you this link. Now, this is gonna take like don't try it now. what happens is, like, I was trying to show the class this morning, and I found out when we are in Zoom, I guess Zoom is taking up a lot of your internet bandwidth, and then when you try to use this, it's like super slow. Okay, once you get off class, and then when you do it on your own, it's gonna work out just fine. Okay, so here, like, just follow the step. Read the instruction. Okay, that's very important. Launch the website I just did. Click on Learn and click on the question mark with the outline area to investigate the microscope parts. So that's what we're gonna do. We're gonna click on Learn. Don't do this on your phone, okay? Find a computer and do it there. And then you can click on the different parts and find the names. Okay, so for example, like what is this? Oh, this is called the eyepiece, also called the ocular lens. Typically 10 times, but also comes in other variants. 10 times is the most common one, okay? 10 times. And so that's called the ocular lens, eyepiece, right? So you check out the other stuff. Oh, that's called the stage, and then you read about it. And then it's the easiest thing. You take a look here, and you're like, okay, eyepiece or ocular lens. What letter is it? Okay, look at the picture. What letters are we going to put over here? Tell me. Did you pay attention? Yeah,
C.
that's right. You put C here. There you go. That's 0.5, okay? And then you do it for all of them. And then you get 4 marks. Okay? You can do that, right? I hope so. That's it for the first part. Objective number 2. Click on Main and return to Main menu. Click on Explore and so on and so forth. So I'm just like, you read it when you do it, right? So like, you're not gonna, you know what step to do, but I've done it so many times I remember the steps. Okay, so click on Main and then click on Explore. And then you're gonna open up this slide box and we're gonna choose Sample Slide, letter E. Okay, it's okay if you don't remember the steps because it's all written in the thing, right? In the lab report. So it's so blurry. What's going on? Well, if you read the description of what to do, you will know, okay, like I have to adjust this. This is called the stage, right? The stage moves up and down and it helps you focus on the specimen. And so we're gonna do the course adjustment. You can see, look at the stage, okay? Look at the stage as I move it. Right? This basically turns the knob on the side and then, you put it into the focus. And then if it's still a little bit hazy, you can fine tune it with a fine focus, and it's much better now. Right? Fine focus, move the stage slowly, right? So you can get it to the right spot, the sweet spot. Course adjustment goes like up and down really quickly. So that's it. This is at 4 times magnification, and what I want you to do is to take the take a screenshot of it. Okay, so for those of you who don't know, if you use PC, there's a something called snip. Okay, just type in snipping tools in the question mark here, and then you open the snipping tool, and you click new, and then you copy this. Okay, you copy this, and then you go to your assignment, and it says all the questions are highlighted in yellow, right? Okay. Center the image and take screenshot for the letter E at 4 times, 10 times, and 40 times. Place your screenshot below like this. Now you just put it there. Obviously, it's too big, so please make it fit. That's all I want. Okay? Don't give me the whole page. Okay? Like, it drives me crazy when people do this. Okay? And then like put this whole thing into the assignment. Like, I look at that, I just get like a little bit unhappy. You don't want me to be unhappy when I mark it. Right? Just the E. Do you understand? Now, some of you use Mac, right? Apple computers, right? You probably have similar function for copying screenshots. If you don't know how to do it, just use your phone. Okay, take a picture of it, and then insert the picture onto into the Word document. Is that okay? Anybody use Mac here? Like, what is the command do you guys have like a screenshot command? Or nobody use Mac here?
Well, I use Mac, but I don't know.
Oh, okay. Jay, is that the Mac command? It says Command Shift 5. Shift
Mine
Command
is Command
3.
Shift 3.
Okay. So try it out, Patricia. It's one of these things. And either way, right? Oh, Command Shift 3 is for the whole screen. Oh, there you go. All right, so here, for people who are watching the recording, and if you're using a Mac computer, okay, thanks to the class, they're saying if you do Command Shift 3, then that takes the whole screen. I do not want the whole screen, okay? Please do Command Shift 5, and it will allow you to select a particular part of the screen. In any case, if you don't know, just Google it, right? Like I'm sure there is some video out there that shows you how to do it. Worst comes to worst, just take a picture with your phone and then transfer the picture onto your computer and then put it onto the document. Okay? And then you gotta do it for like 10 times and 40 times, right? So over here, back to the app, 10 times. But it's it's a little bit darker now. It tends to get darker when you zoom more in, because less light, you're closer to the slide, so you can adjust the light and make it brighter. You can move around, find a good spot that you like, take a screen cap, and then put it in here and here. That's all I want for 3 marks. Okay. Now, what is the relationship between the image, that is what you're seeing through the microscope, and the object? Okay, so here, this is the image, and the actual E of course looks like this, right? So that's the object. This is the image. And this is asking you what is the relationship. We're gonna do it together right now. Tell me, what's the relationship? What's the relationship between the original E and then the E under the microscope?
Letter P and then T.
No, I'm talking about A. What is the relationship between the image and the object? Not asking for the examples yet. Okay, well, I have people who say it's upside down. Anyone else? Do you guys all agree is upside down? Usually I hear another description besides upside down. How would you describe the
It looks
relationship?
like A.
What's that? I'm sorry?
It looks like A.
It looks like A.
Yeah.
Yeah, okay, but then like if I use a different letter, it wouldn't be, it wouldn't be A anymore, right? Like
It's inverted.
it's in, what does that mean? Inverted.
Like opposite.
Opposite how? Sorry,
um,
because I want you guys to really understand what is it that you are explaining, right? Okay, so let me try this again. This is the original, right? I hear a lot of people say it is flipped. Okay, flipped. Right? Flipped is not correct because that's flipped, right? Like that's you can flip it like this, as in like flipping a page of a book, or you can flip it like this, as in like flipping one of those like desk calendars, right? So these are both flipped, but neither of them is the right answer, right? The image looks like this. Okay, so what's the relationship between this and the original? Well, you're right when you say inverted, but what exactly does inverted mean? Inverted means it is rotated 180 degrees. Okay, you can also say it is upside down, but not just upside down, and backwards. Do you understand that? It's upside down and backward. Okay, so that's what inverted means. You take it and you rotate it 180 degrees. Inverted, not inverse. Okay, inverted. So everybody knows the answer now? Now give me another letter that will show this property. Give me another letter.
P.
Yeah, P is good.
G as well.
Sure. Okay. I'll give you some examples that are bad. Okay. O is not good. Why is O not good to show this property? it looks the same, right? Even if you flip it upside down, rotate it, right? Reflect it, it's the O looks the O, you wouldn't know what happens to the image, right? So things that are like symmetrical is not a good choice. Give me something else that's not a good choice besides O.
H.
Capital H, right? I assume you're saying, yeah.
Yes.
Yeah,
Yes.
that's right. S is a perfect example, right? S, if you rotate 180 degrees, it look exactly like the original S. So it's not just like, left-right symmetry, right? Sometimes, rotational symmetry as well. Patricia, you have another one for me that doesn't work or work, whichever you want to tell me.
W,
What's that? I'm sorry?
W or M?
Yeah, does that work or not work?
Doesn't. It wouldn't
Doesn't
work.
work. That's right. Okay, very good. I think you guys get it. But this question asks you for ones that would be appropriate, right? Okay, we want the one that works. All right, so I just gave you the answer for this one. It's 3 marks. Okay, this is just copy and paste work, 3 marks. Okay, so far everybody's getting 100%. Right, this one is just matching. This one is just copy and paste. This one I already gave you the answer. Everybody's 100% up to this point. Okay, good job. Let's move on. Objective number 3. Okay, you're gonna read all this mumbo jumbo. Okay, but I'm gonna try to briefly explain to you. Let's go back to the main menu here. And I'm just gonna click on explore. So we have these little magnifying lens here. The ones here, you can actually rotate it and change which one you're using. They are called the objective lens. Okay, down here is objective lens. Usually, well, not usually, but they come with 3 objective lens. Unless you pay more, then you get extra ones. But like usually there are 3. There's the low power, medium power, and the high power. All right, so over here, you can take a look at what they are when you click on the question mark. I honestly forgot what they are. Let me just sorry, learn, and then let's see what they are. Okay, so here is 4, and then 10, 40, and sometimes it's 100. All right, so the low power is 10, the medium is 40, and then the high power is 100. And what happens is the ocular lens will also have its own magnification. Ocular lens typically is 10 times. So listen carefully. If I am using the 10 times objective, how big is the image going to be? It's 10 from the ocular and then 10 from the objective. Together, 10 times 10 is 100. Okay? If I'm using the mid-power lens, which is 40, what's the total magnification?
$400.
That's right. 40 from the objective, 10 from the ocular. 10 times 40 is 400, right? And just humor me. If you do 100 times, what is the total magnification?
1,000.
That's right. All of you understand? Any question about how I got those numbers? Okay, so you fill it into this chart. Right? Everybody's still getting 100 at this point. Now comes the hard part. But before that, any question about any of the stuff that I talked about so far for the microscope lab? I guess I'll find out on Friday. Let's do the hard part now. All right. When we look under the microscope, you will see like the circle, right? That's actually what it looks like when you look under the microscope. You will see a circle, and then your specimen will show up here within the circle. So this, whatever you're looking at here, that is called the field of view. Okay, FOV. And one of the things we're interested in figuring out when we use microscope is the diameter of the field of view. Diameter. Okay, so that's DFOV. diameter of the field of view. How should we measure that? What do you think? If we want to measure the length of something, what tools do we use? It's not a trick question. Just say whatever comes to your mind. Yes, we use a ruler. That is correct, okay? Now that might sound like, deceivingly simple, but it's not. You are gonna stick a ruler under the microscope, right? Like when my class do it next week in person, that's what they will be doing. They will be taking a ruler and they will be sticking it under the microscope. Okay? Now, let's understand how the ruler works. To your surprise, many people actually don't fully get it. How much is that? What's the length between the 6 and the 7?
1 centimeter.
Exactly, 1 centimeter, all right? What about between two lines? Okay, let me zoom in a little bit here for you. Between these little black lines, how far apart are they? That's right, it is 1 millimeter. 1 millimeter. Okay, so within a centimeter, there are 10 millimeters. Okay, so what happens is you're gonna stick this under the microscope, and then you're gonna look into the eyepiece, right? And you're gonna see what it looks like under the microscope. Because you're zooming in so much, you are going to be seeing the ruler quite up close. And what you will end up seeing is like this. Okay, like you're gonna see the lines like this. Oh boy. Give me a second here. Okay, I'm trying my best here to draw them. So there we go. Okay, you're gonna see the length. And so how far apart is that? What do you think? Based on everything I taught you so far.
1 millimeter.
That's right. That is totally 1 millimeter. You are so zoomed in the ruler that you're looking at the space, right, between two of these lines. Can you visualize what's going on here? Right, like I'm pointing at the screen, so you can't see me pointing. Like over here, from this line to this line is 1 mm. Okay, and from here to here, that's another mm. That's another mm. Right, and over here, like, I mean, I don't really see the next line, but then I have a little bit of gap here. So if I put it all together, right, the diameter, the DFOV is 4 millimeters. Does that make sense? 4 millimeters. Right, so DFOV at, I will say this is let me write it out for you. Sorry. Let's just say this is at 4 times objective. Okay, 4 times objective is how much magnification in total? Do you remember? 4 times objective, the lenses, how much, what's the total magnification?
So it's 40 times 10.
That's right, so 40 times total mag, right?
Yeah.
Makes sense? That's it. So the DFOV, according to the ruler, is 4 millimeter at 40 times mag. Mag is magnification, right? Now, we often do not report length in millimeters because the things we're going to be looking at are so small. So, micrometer is more appropriate. We're going to times 1,000 micrometers per millimeter, and that's going to give me 4,000 micrometers. Okay.
Oh, it's not 3,500.
What's that?
I got 3,500.
But this is a different example than the one in the lab, right? Right. So look, this is 1 gap, 2 gaps, 3 gaps, 4 gaps. That's why it's 4,000 across, right? Yeah.
Okay.
Sorry, was that?
Yeah, I was talking about the lab assignment.
Oh, okay, okay.
Mhm.
Mm-hmm. All right. So like if you use a different microscope, it might be different. So over here, all right, you only have to do the ones that are yellow, okay? A ruler is placed under the microscope and viewed using the 400 times magnification. What is the diameter of the FOV in micrometer? So if you look at this, what do you think? Give me some numbers, guys. Give me some numbers. What's the DFOV?
1,000?
Okay, 1,000. That might be a little bit too short, but let's start with that. Anyone else?
3,000.
3,000. Okay. Well, anyone else? We have 1,000. We have 3,000. Anyone else?
4,500.
What's that? 4,000? Did you say 4,000?
4,005.
4,005?
Yeah.
Okay. Yes,
Just
Patricia.
4,000.
How many, sorry?
4,000.
4,000 for this particular picture? Yeah, okay.
Sorry,
How'd you,
what are we measuring? Just in between the line or the diameter of this?
the
Diameter.
diameter is from here to here, like basically
Yeah, the whole
across, right?
thing.
Yeah,
Yeah.
so how big is it? 6,000. Oh my goodness. So far we have
2,000.
what is that again?
2,000.
2,000. Thank you. 2,000. You changed your mind. Okay. Do you guys understand what is this mark? What is this mark? This is the ruler, right? This is the marking on the ruler. This is another marking on the ruler. What's the distance between two lines on the ruler? What's the distance between them? What did we say? How far apart is this?
1 millimeter, 1,000.
Yeah, 1 millimeter, right? 1 millimeter. Okay, so what about here? Like that is like not yet 1, right? Because I don't see the other line yet. So it's not yet 1, but it's more than half, right? For sure. Right? So like that's like 1.7 maybe. And then there's a little bit here as well. So if I take this and then I add that together, everything put together across, how big are we talking about? What's the distance?
2 millimeters.
2 millimeters, right? Do you guys understand? Right? They want it to be in micrometers, so I times 1,000. I want to know, how did you guys get like 4 and 6? Like which part was not clear? I assure you, you are not the only one. Okay, like I get all sorts of weird answers over the years for this. Yeah, hopefully you know it's 2 now, but I want to understand how did you guys get some of the other answers. So when I explain it in the future, like I can tell people not to do certain things.
So I thought it was like 3,000 because there's like an arrow there, right?
This arrow?
Yeah. And
Oh.
for some reason I thought, okay, maybe like that's one. Like one
You're
centimeter.
like 1, 2,
So
3,
then
4 like that?
no, I just thought the length of the arrow to the circumference of the circle is like 1 centimeter. So then you said the diameter, and then I was like, okay, it looks like 1. So then the total would be like 3, maybe.
I see.
I guess.
Okay, so actually, the arrow here. Oh my goodness, what did I press? The arrow here is a good thing that to talk about. Oh boy, what did I do? Hold on. The arrow is not part of the ruler. The arrow is part of the microscope. Right? It's etched into the lens. It's useful for like when I'm looking at something and then I want to show like another person what I'm looking at. I'm like, oh, look at that cell. That's an interesting cell. But I can't physically point to it, right? So I would move my slide so that the arrow is pointing at something that I'm interested in, so that when the next person looks at it, they know exactly what you're pointing at. That's the purpose of the arrow. Okay. Maybe I'll try to take that out in the future. But are we clear now? Like people who did not choose 2,000, have I convinced you that this is 2,000? Okay.
Try the next.
Sorry, what's that, Patrice?
Oh, that's nice. So let's try the next one.
Let's try.
I don't know
Yeah,
my
let's do the whole
microphone,
thing
sorry.
together, guys. Right? Yeah, why not? Blessing, not to put you on the spot, but can you tell me how you get the 6,000? I'm curious. Like, what was the thought process of that? If you don't mind.
You know, you drew one arrow from the beginning of that of the circle to the other side. So I just
Like this?
yes.
And then?
I just guessed it was just a guess number.
Oh, okay. But do you understand why it's 2,000? That's more important.
Yes,
You
yes,
sure, right? Okay, we
yes,
just
maybe.
we look at the black lines. Okay. Now moving on now. Let me see. Okay. Take out your calculator, guys. Okay, you need your calculator for this next part. Okay, let's do one more, and then we're gonna do some calculations. I hope you all get this right this time. Okay, what is the DFOV in this picture? DFOV at, by the way, this is 4 times objective. 4 times objective means 40 times magnification, right? So what is the DFOV for 40 times magnification? Okay. I am all ears.
$4,500.
4,500. Anyone else?
Micro
Meter, yeah,
meters.
sure. Anyone else? I'm not gonna say if this is right or wrong.
450 micrometers.
Sorry, what was it? Sarah, go ahead.
4,250.
Okay, very precise. I like
Yeah.
that. I tell you, what did you say?
The answer is 450 micrometers.
450?
Yeah.
Like this? 450?
Yes.
Okay. Anyone else?
I mean, that's the final answer.
Final answer? Yeah. Okay. Anyone else here? Well, I'm not, I mean, I will accept a range of answers, okay? Like, I'm not looking for like a specific one, but this is your thought process, hopefully. Yeah, Bibi, you have another number you want to give me?
I'm just, I don't know why, but I thought it was 40 times 10 times 4, which is 1,600.
No. The magnification has nothing to do with the ruler. Okay, you got to remember you are looking at a ruler here, right? This is the ruler, very zoomed in. All you have to do is count the space. Okay, one space. That's 1 mm, 2 mm, 3 mm, 4 mm, and then a little bit, right? A little bit. How much is that little bit? You know, some of you might think that's half. Some of you might think that's like a quarter. Some of you might think it's like, 0.3. It doesn't really matter. I will accept all of them. Okay. So it's 4 point, I'll say 4.3. Okay, 4.3 millimeters. You can say 4.5, it's okay. All right, but I wouldn't accept 4, right? Because clearly it's more than 4, right? It's 4 gaps and a little bit, right? And a little bit. So this little bit, whatever you want to call that. Okay, 4.3. And we're gonna times 1,000 to get to micrometers, right? So that's 4,300. Right, so DFOV at 4 times magnification, 40 times magnification is 4,300 micrometer. Okay, you can keep the 4,500. It's fine. So what if I switch? What if I switch this to a higher magnification? What if I switch it to like 10 times objective? 10 times objective length. 10 times objective means total magnification is 100, right? So what's the DFOV at a higher magnification? So we're going to use this formula. The formula is right here. And if you need to watch a sample calculation, you can click on the YouTube link here. But I'm going to do it right now for you. Okay, here's the formula. DFOV 1 times magnification 1 equals to DFOV 2 times magnification 2. Okay, that's the formula. The starting DFOV is 4300 micrometer. And that is happening at 40 times magnification. I want to find out the new DFOV when we are at 100 magnification. Okay. Now, hopefully you remember what to do in math class. We are going to be dividing both sides by 100, right? So DFOV2 is going to be, you just key this into the calculator, right? 4300 times 40 divided by 100. So the answer is 1720 micrometer. Okay. By measuring the DFOV at a lower magnification, I can use it to calculate what the DFOV is going to be at a higher magnification. You can see the diameter actually gets smaller because we're zooming in, right? When we zoom in, the distance from one edge to the other edge of the lens becomes smaller. Okay, why don't you repeat this process? Okay, let's do this again. But this time I'm going to be doing, let's say we have like 100 times objective. Okay, 100 times objective means 1,000 times mag, right? Let's find a DFOV. Give you a moment to do that. I don't think so. Sarah, could you try that one more time, maybe, please?
Oh yes, now I got it. Why I put it wrong.
Oh,
Why
yeah,
that?
that's fine. That's fine. Actually, you know what? Maybe you're right. I don't know. I didn't do it yet.
No,
I was gonna
no. I
ask,
think I'm
Yeah,
right.
you're right. Yeah,
I
okay.
think she's right too. Yeah.
Okay, yeah. Sorry, because I used different numbers
No.
this morning, and I'm just like probably remembering
No,
it.
it's because I forgot I already put it in 1,000. That's
Okay,
why I thought that I forgot to put like to multiply by 10, but no, I
right.
did it.
Okay, great. Thank you. Sorry. Let's do it together. So DFOV1, I can use the original. Okay, and then times the original magnification, as in like the original here, right? You could use the orange one, it's fine as well, doesn't really matter. The formula will work. And then here I will have DFOV number 2, like the new one, times the new magnification, which is 1,000. Okay, so again, you will divide both sides by 1,000, and you can plug it into the calculator. 4300 times 40 divided by 1,000. That is going to be 172 micrometers. Okay, that's all. So you will have to do the calculations here. Right? This is a field, and then it shows you the marking of the ruler at 40 times magnification, and then I want it to be at 400 times. Okay, you can do the math on a piece of paper, take a picture and upload it. You can type it in as well. I think uploading a picture is probably easier. Typing it is probably going to be a lot of work. And then this question, number 7 is a continuation of number 6. Okay, so you got to make sure your calculations are right in number 6 before you do number 7. These are onion cells, and the question is asking you to find out like how long one of these things is. Okay, how long one of these things is. So I'm going to show you how to do it with another example. Okay, let's say yeah.
I'm so sorry to take you back. I don't understand this calculation. I understand the later part, but the beginning, how did you get 4.3?
How do I get, like at the beginning?
Yes.
Yeah, so remember we are just counting the empty spaces, right? We're just
Yes.
counting empty spaces. So you understand how there are 4 gaps here? 1, 2,
Yes.
3, 4, yeah. And
Yes.
then this little one, this little gap here.
Okay, is it 3?
Yeah, like yeah, how much is this? How much do you think this is? This little gap here. Like it's less than half, right? It's
Yes.
probably less than half. So that's why I say 0.3. Like it's okay if you think it's 0.4, right?
Okay.
It's even okay if you think it's 0.5. Like I'm just saying this is not like exact science, right? Like it's up for interpretation. So I'll accept a range of answers. Okay. What I will not accept though, like if I show you this picture and you tell me it's 4, then it's wrong because it's clearly more than 4, right? I will also not accept 5 because it's, clearly less than 5, right? So anywhere between 4.2, 4.6, right? That's okay. Is that better?
Yes, thank you.
Okay. So this is in case you don't know, these are red cells, and then we got some white cells here. Right? Let's say I want to know how big is a white cell. Okay, like what's the width of it? Okay. And let's just say this is not real. I'm making this up. Let's just say this is at 1,000 times magnification. Okay. Assuming we're using the same microscope as the calculation here, because if you switch microscope, the field is going to be different. Okay, the diameter is going to be different, and you have to measure it again. So based on our calculations, okay, we figure out all the way across is 172, right? That's our calculation. So how big do you think one of these cells are? Well, what you gotta do is you gotta do some estimation. Okay, let me just try to make this a little bit brighter so it shows up. So like okay, that's one cell here. Let's say I can fit another one there. All right, 3, 4, 5. All right, I can fit around 5 of them. Right, five cells across. Right, just visually, approximately. So that's why I'm going to take 172, which is like the whole diameter, and I'm going to divide it by 5 cells. Right? What's that? That would be
34.4.
Thank you. Okay. Micrometer, right? Does that make sense? That's how you do it. And then there is the last part here. Last part is pretty straightforward. You will go, these two are very straightforward. Okay, you can figure that out yourself. And here it asks you to take a screenshot of a cell in anaphase for onion root at 400 magnification. I'll see if this works now, because like I said, it's really slow when I'm running Zoom. Let me see. Explore. Plan slide, onion root. All the instructions are on the oh, it actually works. So like you can focus it a little bit. Obviously at 4 times magnification, you can't see any of the cells. I mean, I can see the nuclei, but I can't see the cell, right? So you got to zoom in a little bit. Still can't really see it. Maybe you want to zoom in a little bit more, right? So now we are at 40 times objective, which means a total of 400 magnification, right? which is what the question is asking you to do, 400 magnification using the 40 times. And I have to find something with an anaphase in it. Okay, so you gotta go around, you can turn up the light a little bit. Okay, and then you gotta go find the anaphase. Okay, I'm not gonna tell you where it is, right? I see it already. And then when you find the anaphase, you're gonna take a picture of the anaphase. This is, again, using the screenshot function, right? Don't just copy the whole thing and make me find the anaphase. Okay, like I'm not gonna give you marks for it. If you capture the whole thing, then you circle the cell that you think is an anaphase. This is not anaphase, okay? I'm just saying. Like if you're gonna take a whole picture like that, make sure you circle the cell that is anaphase. Okay. Otherwise, just like copy the exact cell that you want, and then you paste it in there. Do you understand? Okay, so you have to go around and find the anaphase, right? Okay, this what phase is this anyways? Do you know? Does anybody know what phase this is? The one I'm like pointing at? The mitosis stuff? Okay, that's metaphase, right? That's all lined up in the middle, right? Okay, so if you need a refresher on what the phases look like, that's what you're gonna do. And then there is just one table here. I'm sorry, there's another one here that asks you to look at the interface and then describe the difference you see between plant and animal cells. You can do that. And then there's a chart here. You know, we need a couple of things here. Capture a cell that is undergoing the indicated mitotic phase. Okay, so I need a cell that is in prophase, metaphase, anaphase, and telophase, and you can put them all here. Okay, a lot of people forget to do this part and they lose 2 marks. And then describe the location of the chromosome. Okay, like where is it? Like, is it in the middle? Is it like moving away towards the end of the cells? Where is it? Right, and then describe the spindle fibers. You might not be able to see the spindle fibers clearly. You won't be able to see it for the plant cells for sure, because plant cells don't have spindle fibers, right? But maybe let's see if you can see it with the animal cell. I don't remember. Animal cell, you know, they have the different phases here already, right? So it's easy for you to find them. Yeah, it's gonna take some time to load. Again, because we are on Zoom, right? So you can focus it a little bit, and obviously you can't really see it at this magnification. You gotta go up higher, and it's gonna load again. Maybe it's faster if I turn off my video. Nope, it's not doing anything. Okay, and then you gotta go up one more time. Okay, just wait for it, be patient. Okay, yeah, so you can actually see the spindle fibers. You see that? Okay. These are the chromosomes in the middle, and then these are these lines. They are the spindle fibers, right? So you want to describe them. Okay, like what are they doing, right? You can also look at the notes, right, to help you see what's the proper way of describing it. And that's it. Okay, honestly, this lab is not difficult. I did most of the work for you already. All you have to do is, you know, go through it yourself. Maybe you want to do it now when it's still fresh in your head, right? Okay, so fill in everything, submit it on eCentennial. This thing is due Friday midnight. No class on Thursday. You will just work on your test, sorry, your quiz and your lab, and I'll see you again next Tuesday for the synchronous class. Okay, that's it. Good luck. Thank you for coming. Have the good week, guys. Bye-bye.